This is the second part of 4 parts for Chapter 4 in the micro lectures. In this one, we're going to look at protein and gene regulation. So how can we regulate a protein and how can we regulate a gene? Let's get started. So first, in an activated gene, we have something called messenger RNA. Messenger RNA is the mirror image of the gene which is made. So you have the gene from the DNA, and the messenger RNA just reads that information from the DNA and becomes messenger RNA. Messenger RNA will then migrate from the nucleus into the cytoplasm, and then its code is read by the ribosomes. Ribosomes and its ribosomal RNA are little granules and enzymes that are able to read the codon that's on the messenger RNA. The ribosomes then signal a transfer RNA to deliver an amino acid that is the exact same amino acid that that code calls for. Then the ribosomes assemble the amino acids in the order directed by the codons, or those 3 bases, on the messenger RNA. And they add them together by a peptide bond. So the process of protein synthesis is always DNA, messenger RNA to protein. The 2 steps are transcription and translation. And these 2 steps are the 2 steps that most students and most teachers struggle with teaching and students struggle with learning. Because transcription and translation are so close, when you read those 2 words on the test, you are very likely to get them mixed up. So transcription is DNA to messenger RNA. Think of it if you're writing or taking notes. You're transcribing. So you're just copying what's being said or what you're reading from. So the same thing here, transcription, just taking the notes from DNA to messenger RNA. And this occurs in the nucleus because DNA cannot leave the nucleus. Translation, on the other hand, is messenger RNA to protein. So it's translating the codons on the messenger RNA into amino acids of the protein. Because it's a different language, that's why this is called a translation. And most of this occurs in the cytoplasm. And then 10 to 15% of the proteins are going to be synthesized in the nucleus. This is why when I originally said in the other micro lecture that some of that stuff was called junk, well, they're able to find out that some of that stuff is actually very important. So some of the proteins get synthesized in the nucleus to be used inside the nucleus. So let's look at this. Steps in protein synthesis. First, the gene has to be turned on. Something needs to activate it. And it needs to remove the histones, and the DNA needs to uncoil. Then the DNA needs to separate into 2 separate strands. And this is what it looks like when they're separated. That's what it looks like when they're paired. Then you have an enzyme. So an enzyme assembles nucleotides in a single strand of messenger RNA. The complementary base pairing matches the DNA nucleotides. And when there is a T, you switch that out with a U. So if it's A to T, it would be A to U. And C to G or G to C remains the same. In a series of 3 RNA nucleotides, that's what's called a codon. And again, each codon is going to code for a specific amino acid. Messenger RNA can leave the nucleus through nuclear pores. Then, at the ribosome, in the cytoplasm, codons of messenger RNA bind to anticodon triplets of corresponding nucleotides on transfer RNA. So this is your messenger RNA. Anticodons, well, first, ribosomal RNA will come here and guide these 2 to come together. But you can see A will go to U, A will go to U, C to go to G. The codon is going to be on the messenger RNA. The anticodon will be on the transfer RNA. And each anticodon is going to be carrying a single amino acid. And then ribosomal RNA of the ribosome strings together the amino acids. And they're added together by a peptide bond. So a protein is just called a polypeptide because it means many of these bonds. And if you look at this again, they separate, an enzyme holds them together, they read, and then it eventually makes a protein. Transcription. So now let's just take a closer look at that first phase, because that explained both transcription and translation. So DNA is too large to leave the nucleus and participate directly in the cytoplasmic protein synthesis. So to do this, it needs to make messenger RNA. That process of DNA to RNA is called transcription. That enzyme I kind of showed you before is called RNA polymerase. And RNA polymerase is important because that's the enzyme that binds to DNA and it allows it to unwind and starts to assemble the messenger RNA. The base sequence always begins where TA or TAAA or a bunch of A's and a bunch of T's. And if you kind of go back to that first micro lecture, you might remember why a bunch of A's and T's would be a place for it to start. If you go back, there are two single hydrogen bonds between A and T. So if the corresponding signal to this would be A, T, A, T, A, T, that means this is the weak part of that gene. And it just happens to be at the beginning. It doesn't happen to be at the beginning. It is at the beginning because this is where it always begins. It's the weakest part where this RNA polymerase can bind. RNA polymerase opens up the DNA helix of about 17 base pairs at a time. It reads base pairs from one strand of DNA, only one. It makes corresponding messenger RNA. Where it finds a C on the DNA, it adds a G. Where it adds an A on the DNA, it adds a U. Next step, RNA polymerase rewinds the DNA helix behind it. The gene can be transcribed by several polymerase molecules at once. The terminator is the base sequence at the end of the gene where it signals the polymerase to stop. This is called pre-messenger RNA because it's still going to be in the nucleus and it's still way too long to leave the nucleus. So you have some things called exons and introns. Exons are portions of the immature RNA that will eventually exit the nucleus and translate to a protein. Introns, on the other hand, are going to be staying inside the nucleus and must be removed before it leaves the nucleus. It brings us up to this one term that didn't exist when I was originally in college called alternative splicing. This is by removing introns by enzymes and splicing the exons together into a functional RNA molecule. So one gene can code for more than one protein. We'll look at that later. So let's kind of look at this. Kind of already went over this, where one strand is going to have a strand that corresponds to it. RNA polymerase is going to unwind it, and then this is where the codons are going to be, red. Then you're going to have the transcription of the messenger RNA where it reads, where there was a G, a C, a G, a C, a G, C, a G, an A, a U, a T, an A, and each three at a time will correspond to a codon. And you can see the first codon, G, G, or C, C, G would be the next codon until you have a whole strand of pre-messenger RNA. Once it reaches the end and RNA polymerase detaches from the DNA, you have a messenger RNA strand. And that's each step so far. So now we need to leave the nucleus. So introns and exons. So you have exons, which are the dark green exons, and you have the light green, which is introns. What happens is that there's enzymes that remove these pieces that stay inside the nucleus, and then you can combine the exons to form a specific part of messenger RNA. before we leave, we did say alternative splicing. You could splice A to E to I, and that can be another type of messenger RNA. It can be E, A, C. That can be another type of RNA, and it's enzymes that are controlling this. So all of these splices can be combined in different combinations. That's how one gene can code for multiple proteins. So now we're outside the nucleus. Now we need to translate those codons into amino acids. So translation is that process that converts the language of nucleotides into the language of amino acids. Ribosomes translate the sequence of nucleotides into sequences of amino acids. They're the ones doing it. Mainly going to occur in the cytosol. Remember that 10 to 15% happens in the nucleus because those enzymes that are helping with DNA and RNA synthesis, they're actually proteins also. They consist of two granule subunits, one large and one small. We're not going to get into those, but we're going to look at the three phases of RNA translation: initiation, elongation, and then termination. So the messenger RNA molecule begins with a leader sequence. It's always going to be a start codon with AUG, or there's probably, I think there's 5 recognized start codons. There could be more now. And what happens is one of those start codons goes to the ribosome that's going to start with the first amino acid called methionine. So every protein in your body begins with methionine because that's what codes for that. That transfer RNA has the anticodon for A, U, and G. This is the transfer RNA with its messenger RNA. It's just a long folded part of RNA, and at the end it has an area where it can accept an amino acid, and that's how the anticodon can read the codon on the messenger RNA and bring the amino acid to the ribosome. We're not going to get over to the L end or whatever. We're just going to say anticodon's on one end reading the messenger RNA. The other end is going to bring the amino acid. So some imprecision in codon-anticodon pairing. It takes 48 different transfer RNAs to pair with 61 different codons. Ribosome binds and holds transfer RNA with its specific amino acid. And then the large ribosomal subunit contains an enzyme that can form the peptide bond together. And then once it kind of reads the first one, it sends it to the next step, and then eventually it'll release it. So let's look at this process. So the first one comes down. This is the codon. That's the anticodon, and that's going to code for methionine. It goes there. It goes into this position, and then the next amino acid gets brought to the codon by the anticodon, and then the large ribosomal subunit will add a peptide bond to it. That'll shift everything over to the next thing. So now this one right here goes to the middle slot. This slot right here will push this one off. This will go and rebind to another free-floating amino acid in the cell. And then the next one gets brought. And this process continues until it stops. So initiation was the first one. This is how it gets longer. And then finally we're going to look at how does it terminate. It terminates once the small and the large subunits become apart, and then you have a completed peptide of either 40 amino acids long or a couple hundred of thousand amino acids long. And that's the whole step of initiation, elongation, and termination. So let's review peptide formation. Starts with the DNA. This is going to be transcription. And then right when it goes into messenger RNA and leaves the nucleus, this is going to be the process of translation. And it's translation because it's codons into amino acids. So the second part of this lecture, protein processes and secretion. How do we get proteins out? How do we make all these different types of proteins? So just because a protein is made on the ribosomal RNA, it doesn't mean it's done. Because once the amino acid is sequenced, it has to be assembled, it has to be folded, it has to be coiled. So for it to be functional, an enzyme is just a type of amino acid sequence, and they have to be folded in such a precise way that if it isn't, it won't bind properly. So you have other chaperone proteins that are in the nucleus, or sorry, in the nucleus if they're making the ones inside the nucleus, but outside the nucleus you have these chaperones that are older proteins that pick up new proteins and guide the new proteins in folding into proper shapes. They help prevent improper association between different proteins. It also called stress proteins or heat shock proteins because these are produced in response to heat or stress. But what these do, they help damaged proteins fold back into their correct shapes. And the process is actually pretty complex. You can have free-floating ribosomes, or you can have ribosomes on a endoplasmic reticulum. If it's on the endoplasmic reticulum, it's called rough ER. And because they have these channels, these cisternae we talked about later, or before, these allow the proteins to go in and fold in a specific shape. Once they're folded in a specific shape, they get packaged, and then they get sent to the Golgi complex and the Golgi apparatus so it can be folded properly. Once they get modified, they can have lipids, they can have fats, which is the same thing. They can have minerals added to them. Once they kind of have everything built around them, just like a car in a factory, they get shipped out into Golgi vesicles and then can be secreted into the blood, secreted into the or the cytoplasm, or into the cell to be reused. They can be added to the cell membrane, or they can be released into the outside of the cell. So, like I said, proteins used in the cytosol are likely to be made on free ribosomes in the cytosol. Because they're smaller, they're just going to be there. Quick ones, there you go. Proteins destined to be packaged into lysosomes or secreted from the cell are assembled in the rough ER and sent to the Golgi complex for packaging. They do this because they're much more complex. So there's not much to really think about that. You'd always, if you want to go and clean your car, you can do it outside. But if you wanted to do a full body clean and everything else, you might want to bring it inside. You might need to shampoo the carpets and stuff. You might need a factory or some special place to do all that. And proteins assembled on the rough endoplasmic reticulum surface threads itself through the pore of the ER membrane into the cisternae, and then they just kind of have post-transitional modification. They can remove some segments, they can fold the protein, stabilize the protein, they can have disulfide bridges, they can add carbohydrates. So pretty much everything I just said, they just make them more complex. And nothing's in red, so you don't need to know anything more than the proteins made in the cytosol are pretty basic. The ones that are made in the rough ER are more complex. The ones that have to go to the Golgi apparatus are even more complex. And then that's what I'm kind of saying here. Once it goes into Golgi, it becomes minerals, fats, all these other things can be added to it. These can be turned on and turned off. They changes from minute to minute, hour to hour, day to day. Some days your cells might be producing this one protein. Other days they might say, we don't need that. They do it with hormones, they do it with a bunch of other different types of things. You can have genes for hemoglobin. If you go to a higher elevation, your genes for the hemoglobin are going to start making more hemoglobin. If you're digesting more food at a certain time, enzymes for digestive system will start to be turned on. And you have mechanisms for them to be turned on. One example for this is going to be a mother giving birth to her first baby. The hormone prolactin stimulates cells of the mammary glands to begin synthesize components of breast milk, including protein casein. The prolactin binds to receptors, a pair of proteins in the plasma membrane of the mammary cell. So it pairs with the plasma membrane. This receptor triggers activation of a regulatory protein. So an activator in the cytoplasm. This regulatory protein moves into the nucleus, binds to the DNA near the capsaicin gene. Then this binding enables RNA polymerase to bind to the gene and then transcribe it, producing the messenger RNA for capsaicin. Capsaicin messenger RNA moves into the cytoplasm and is translated by ribosomes on the rough endoplasmic reticulum. The Golgi complex packages capsaicin into secretory vesicles. The secretory vesicles release the capsaicin by exocytosis and it becomes part of milk. So prolactin binds to it. It uses energy. The regulatory protein moves into the nucleus because it's small. It goes through the nuclear pore. It binds to the part of the gene that codes for the capsaicin. RNA polymerase starts transcribing the gene. Then you have the messenger RNA. The messenger RNA makes the amino acid sequence for that, and then it gets packaged into eventually capsaicin. And finally, cells that synthesize glycogen, fat, steroids, phospholipids, pigments, and other components. There's no genes for these. So because they're not proteins, you don't have a gene for them. So these are under indirect genetic control. They're produced by enzymatic reactions. These enzymes are the proteins that get to be encoded by genes. So because it's indirectly, these enzymes have to be made that can then make these. So an example, testosterone is a steroid. A cell of the testes takes in cholesterol, enzymatically converts it to testosterone. Only occurs when genes for the enzymes are active. So it might seem like, yes, DNA only codes for RNA, RNA only makes proteins, but those proteins are able to make every single thing our body needs. That's why they're so important. Genes may greatly affect complex outcomes such as behavior, aggression, and sex drive. So hopefully after this lecture you understand how important they are, and by knowing the building blocks of them will help you better understand yourself and your behavior.